Prince PhD thesis defense. A little background before we begin. Lauren is a native Floridian. She finished her undergraduate degree at the University of Miami in marine science and biology in 2015, which to me seems like yesterday, but that's just me, I guess. It seems like literally yesterday. She went on to get a Master's of Science degree at the University of the Virgin Islands in 2017, so 2 years. That took her in Marilyn Brandt's lab, working on competitive interactions between corals, sponges, and seaweeds, and she subsequently published that work in Marine Ecology Progress series. Now, I was fortunate to meet Lauren at the World Sponge Conference, yes, there is such a thing, in Galway, Ireland in June of 2017, so the summer of 2017, and was even more fortunate to recruit her to my lab, where she began in January of 2018. Lauren has done an amazing job in the 5 years that she's been here, having already published 2 of the 4 chapters in her thesis. She's made conference presentations at the Benthic Ecology Meeting, the International Coral Reef Symposium, and just last month at the World Sponge Conference, which comes around every 3 or 4 years, in Leiden, the Netherlands. In my 32-year career, I have never had a student as technically proficient or as artistically inclined as Lauren. Her thesis work became the foundation for a recent NSF award to Dr. Strangman, Mead, Johnson, and myself that will continue to keep us and a new crop of students busy for the next 3 to 4 years. And as a testimony to her abilities, Lauren has already lined up a postdoctoral fellowship beginning in the spring to return to the University of the Virgin Islands and the lab group of Dr. Tyler Smith. So with that, I would ask the Zoom group to please mute yourself. And Lauren, take it away.
Thank you, Joe, for that nice introduction. And thank you all for being here today, either in person or over Zoom. I'm really excited to be sharing the culmination of this almost 5 years of work that I've been doing here at UNCW. So it's only been 6 months since my prospectus seminar, where I talked about these 3 main parts of my research. And I'll be going over those again today, but I'll also be giving an update, especially with some new results for part 2, and then also adding some more details and context throughout to kind of hopefully complete the story. To start, our research focuses on Caribbean coral reefs, where, as some of you know, sponges are very abundant. And so to demonstrate this, from this survey of about 70 Caribbean sites, sponge cover averaged 16%, which was comparable to the cover of the corals that build these reefs. And sponges are also only increasing in abundance as corals continue to, unfortunately, die out in the Caribbean. Not only are they abundant, but sponges are also really good at pumping seawater through their bodies, and this gives them a pretty important role from an ecological standpoint, because as they pump all this seawater, some sponges can consume dissolved organic carbon, or DOC, from it. And DOC is a fraction of total organic matter that is operationally defined by what passes through a filter, typically with pore sizes either of 0.2 or 0.7 microns. And this separates the DOC from the particulates, or POC, that are retained on the filter. And DOC is a part of dissolved organic matter, or DOM. So as sponges are eating this DOC, they are changing the chemical and molecular composition of DOM. This abundance of sponges filtering a lot of seawater and consuming DOC from it has led to the suggestion that they are important for cycling carbon on coral reefs, because as they consume this dissolved food source, they can transform the unrespired fraction of that carbon to something like biomass, or maybe even detritus, that can be used by higher trophic levels on the reef, and this retains carbon in the system. And this is something called the sponge loop hypothesis, which has gained a lot of popularity and also been widely accepted in the past 10 years. This is also a diverse group of animals, and the diversity of sponge species can be divided into functional groups. For example, based on their morphology and preferred habitats, or based on whether they produce chemical defenses, or based on the abundance and diversity of symbionts that they host. So this separates the high microbial abundance and low microbial abundance species, so HMA and LMA. And there are likely significant differences in how these groups use and transform carbon, specifically the types of carbon they consume, and then how fast they transform that to biomass or detritus, if at all. And I've denoted the 3 parts of carbon here in bold, and they are the focus of each of the 3 parts of my research that I'll now be going over one by one. So starting with part 1, where we cover the uptake from the DOM, and this was the focus of the first 2 chapters of my research. So here we are focusing on this section of the carbon cycle, looking at the types of DOM that sponges take up and some sources of this DOM. And with respect to the source of carbon consumed by sponges, if it is from the DOM, we know a lot of this is coming from primary producers. So things like phytoplankton, corals, benthic algae, as well as benthic cyanobacterial mats, or BCM. And we don't know if sponges take up the DOM produced by BCM, but this is important to find out because BCM are becoming more abundant in the Caribbean. Otherwise, sponges appear to prefer algae over coral-derived DOM, and we know these are compositionally distinct. But beyond this basic understanding, we don't know much about the molecular composition of DOM on coral reefs, mainly because we haven't had the techniques to be able to characterize it until relatively recently with new high-resolution mass spectrometry techniques. And these approaches can help us resolve the low molecular weight variant of the DOM, which represents a lot of this labile material that is ecologically important because it may be consumed by heterotrophic water column bacteria or sponges, but otherwise it's not a very common food source for metazoans. So it's a fraction of carbon that might be lost from the reef if not consumed by sponges. But this fraction of the DOM may be more accessible to the HMA species that have the symbiotic microbes that we know can take up these small molecules. And some studies have reported DOC uptake only by HMAs, while other studies have reported DOC uptake by both HMAs and LMAs. So there are some inconsistencies here. DOC uptake by LMAs may be possible if the sponge cells themselves can take up DOC, but the DOC taken up by LMAs may also be part of the larger colloidal fraction of the carbon. And some inconsistent findings may be due in part to the use of different filter sizes to define DOC. So whether you use a 0.2 or 0.7 micron filter. And with these new high-resolution mass spectrometry techniques, we can focus in on that low molecular weight fraction of the DOM that is abundant and important in these ecosystems. And when I talk about high-res mass spectrometry, it's in the context of this metabolomics workflow, which refers to the high-throughput technique that we use to profile metabolites in a sample. And we use untargeted metabolomics to profile all of the metabolites, known and unknown, in that sample. So our objectives for the first 2 chapters of this research were to use untargeted metabolomics to compare the seawater DOM before and after processing by sponges from samples collected on the reef and in the lab. And we wanted to see the origins and types of DOM taken up by sponges, compare HMA and LMA functional groups, and then test our hypothesis that BCM are one potential source of DOM that sponges take up. Our study site was in Caribou Key, Belize on the Mesoamerican Barrier Reef, and we collected our samples from the forereef that was right next to the field station. And for the first experiment, we collected seawater going into and then coming out of sponges on the reef, so in their natural habitat. And we refer to these as in-current and ex-current samples, or just in-ex. For sampling, 2 divers each held a syringe on either side of the sponge wall and collected seawater simultaneously into these foil bags, which gave us a snapshot of the seawater at the moment that it entered and the moment that it left the sponge. We also sampled 3 each of common HMA and LMA species to get that functional group comparison. Then for the lab experiments, this was chapter 2. We started by collecting a sponge and some BCM from the reef and bringing those into the lab. And then we incubated the BCM under light for 2 hours and then transferred that seawater to a jar with a sponge that we then incubated in the dark for 2 hours. And in 2020, we ran 5 trials of this experiment using the HMA species, Cestospongia muta. And the BCM that we used was this consortia of species that included Morea producens, and this used to be called Lingbia majuscula. And at the start and end of each incubation, we sampled seawater. So here are the different sample types that we collected. Then, to analyze all of the seawater samples from both our chapters 1 and 2 lab and in-ex experiments, we started with solid phase extractions in the lab at Caribou Key. Then we brought those extracts back to UNCW, where we analyzed them using LCMS, and then in R to make a list of mass features, where each feature represents an individual compound in the seawater. And then we used a variety of stats to compare the relative abundance of those features before and after processing by sponges and BCM. So now I'm going to show our results from our chapter 1 in-ex experiment, and we published this last year. To start, we used a principal component analysis to look at the overall changes in the DOM based on the relative abundances of the 2,100 features that we found in the DOM. We used arrows to connect the in-ex samples of each individual, and the colors of those arrows represent the species of that individual. What we found was that the LMAs did not appear to make any consistent changes to the DOM, but the arrows representing the HMA individuals are all moving from left to right along the PCA, showing that they did make some consistent changes to the DOM. And we think that the most parsimonious explanation for this distinct HMA-LMA difference is that the HMA microbiome played a part in changing the DOM. And then we looked at the individual features underlying these larger changes and found that they were driven primarily by what looked to be uptake by the sponge. And this is shown in this volcano plot, which plots each feature based on its magnitude and significance of enrichment or depletion. So here you can see the 130 depleted features compared to the only 11 enriched features. So the HMAs were taking up more DOM than they were putting out. Interestingly, these HMAs took up a lot of features that had chlorine and bromine, so they were halogenated. In fact, they took up a disproportionate number of these features, which was interesting and raised a few questions to us. So first, what could the sponge be using these features for? With the help of its microbiome, could it be removing the halogen and using what's left over as a source of food? Or could it possibly be recycling these secondary metabolites or these metabolites into their own secondary metabolites, so things like chemical defenses or signaling molecules? Also, where could they be coming from? One possibility are those benthic cyanobacterial mats, or BCM, which we know are prolific sources of these chlorinated and brominated toxic metabolites. And this is why we singled out the BCM as the potential source of DOM for our lab experiments, which brings us to those results. And just as a reminder, here are the different sample types that we collected. So first, we were interested in seeing how bulk DOC changed during the experiment. And we found that DOC increased during BCM incubations and then decreased during sponge incubations, almost returning back to its initial concentration. And then we made another PCA, and what this showed was that all of the blue arrows are pointing up and all the orange arrows are pointing down. So it looks like the BCM are changing the DOM in some way, and then the sponges are almost reversing that change. And then we found about 1,100 features that were significantly enriched during BCM incubations and depleted during sponge incubations. So these might represent metabolites that are made by the BCM and then taken up by the sponges. We've identified a few of these features, including this caribomide, which was originally isolated from a Lingbia bloom on Caribo Key. And here are 5 compounds that we've putatively identified so far, and notably the 3 on the bottom are all halogenated. So then we wanted to see if there was any overlap between our in-ex and lab experiments. So we looked for what we call recurrent features, defined as any of those 1,100 BCM-enriched sponge-depleted features from our lab experiments that were also depleted by sponges in the in-ex experiments. So we know that they are present in the lab and on the reef, and also appear to be taken up by sponges in both cases. So they seem to be ecologically relevant, but we don't know what they are. And an example of one of these is feature 440. And we found these recurrent features by making a library of fragmentation spectra from our lab experiments. And then you can think of each spectrum almost like a barcode that we then use to scan against our in-ex data to look for matches. And then we plotted the relative abundance of that matched feature in the in-ex samples. So here are the in-ex abundances for feature 440 for the 2 HMA species that we sampled in 2019. And super interestingly, this feature was only depleted by Muta and not at all by Tubulata, the other HMA species. So this is showing some species specificity in the uptake of this DOM, which might be due to, for example, differences in the microbiome between these 2 sponge species. We have several other recurrent features that were depleted by all of the HMAs that we sampled, but notably these features were never depleted by LMAs. So now just to quickly summarize, HMAs were the only species to affect the low molecular weight DOM. And this might be evidence of a function of the HMA microbiome if forming these new symbiotic relationships provided access to new types of resources or compounds. It also might partly explain the divergence of HMA and LMA species. Also, if this ecologically important fraction of the DOM is only accessible to HMAs, then this raises the questions of relative abilities of sponges to cycle carbon back to the benthos, if not all species can complete the loop, or if different species recycle different types of carbon. And then the HMAs depleted these really interesting metabolites, including those that were halogenated, which made us wonder what the source of these metabolites might be. And we found some preliminary evidence that BCM are at least one source of these metabolites. Additionally, we know that sponges take up DOM as a source of food, but they may also be tapping into this resource as a source of secondary metabolites like chemical defenses. And in support of this, there are some uncanny similarities in the defenses produced by BCM in sponges. So it's possible that sponges are scavenging some of this stuff from the seawater. And this is really only a theory for now, but I think it's a really interesting question for future studies, because if there is some exchange of these or other types of DOM between sponges and primary producers like BCM and algae, then this could be helping both groups and making them more competitively dominant. And then by doing this, this could harm whatever remains of reef-building corals by basically compounding the negative effects of what else they're already going through. And this is something called the vicious circle hypothesis. And I think it's another good question for future studies to look at to see what kind of exchanges are happening between these groups. So these 2 chapters raise many new questions about how these organisms use DOM, and these metabolomics techniques provide a new level of resolution that really hasn't been seen in studies of coral reef DOM until pretty recently. And these tools can help us identify real compounds like caribomide, but I think there's also a major value to cataloging the full range of DOM, even the unidentified stuff, because this can act as kind of a fingerprint of different benthic groups that we then can use to trace DOM from source to sink, similar to what we did with our recurrent feature classifications that allowed us to trace DOM from BCM to sponges on the reef.